Welcome to my course on microbial biotechnology. In this module 1, we are going to study about the introduction and principles of microbial biotechnology. So, the module is divided into various lectures, and this particular lecture is divided into 4 different sections. In the first section, we will be studying about the introduction to microbial biotechnology. In the second section, we will be studying about the principles of microbial biotechnology, and then in third, we will be studying about the contributions of microbial biotechnology, and in the last section, we will be studying about some of the emerging concepts and issues in microbial biotechnology. So, let us start with the introduction to microbial biotechnology. So, microbial biotechnology is a multidisciplinary science, and various people define it in various ways. For our purpose, we will be defining this as a multidisciplinary field that integrates principles from microbiology, molecular biology, genetics, biochemistry, and engineering to exploit the metabolic and genetic capabilities of microorganisms for various applications. Lately, the applications of artificial intelligence and synthetic biology have brought new vigor to the discipline. This also we will discuss towards the end of this lecture. As you can see in this particular figure, microbiology is a multidisciplinary subject, and it is getting contribution of knowledge from various domains on the one side, which includes some of the basic science subjects and some engineering subjects, and together they are used for developing technologies for application in various fields like industry, agriculture, medicine and environment. So, briefly we can tell that microbial biotechnology is an applied field that focuses on utilizing microbes for industrial, medical, agricultural and environmental processes, making it essential for advancements in biotechnology and related disciplines. Now, let us examine the scope and objectives of the two different subjects, microbiology and microbial biotechnology. Firstly, if we start with the objectives, you can see that the objective of both the subjects are different. Microbiology, being a basic fundamental science, tries to expand the understanding of the basics of microbes, their behavior and impact, while microbial biotechnology, which is more of an applied discipline, try to leverage this understanding of microbiology to create new technologies or improve existing processes and solve real world problems. Now, when it comes to the scope and focus, microbiology is primarily concerned with the study of microorganisms, including their physiology, classification, ecology and pathology. It aims to understand the fundamental biological processes and interactions of different microbes. While microbial biotechnology applies the core principles of microbiology, then other subjects like genetic engineering, as we have discussed in the earlier diagram, to develop technologies and processes that harness microbes for practical applications such as producing bioproducts in agricultural, medical, industrial and technological contexts. Now, the approach for these two subjects are also a little bit different. Microbiology uses observational and experimental methods to study microorganisms, while microbial biotechnology employs genetic manipulation, metabolic engineering and process optimization to develop and implement microbial technologies. Although this course is focused on microbial biotechnology, we will be discussing a lot about the fundamental aspects of microbiology and some related disciplines, so that we can understand how that knowledge is harnessed for its application in microbial biotechnology. So, once again, let us try to understand microbial biotechnology in a broader way. It is a multidisciplinary field, as we have already told you. It is also a very dynamic field. So, it has evolved over the time. Microbial biotechnology is not a new discipline if we look into the historical development. Prior to the advent of gene manipulation techniques, microbial biotechnology was largely shaped by traditional practices which were centered on fermentation and early bioremediation. And we can see here how these two processes over time has evolved. For example, in the fermentation process by traditional methods, we already have household items that we use daily. For example, fermented foods like yogurt, and then wine, beer, bread and cheese, and these exploits the practical uses of microorganisms for food preservation, flavor enhancement and nutritional benefits. We have to understand that these kind of traditional microbial biotechnology do not use any genetically engineered or genetically modified microorganisms. If we speak about early bioremediation process, it particularly focused on harnessing naturally occurring microbes to degrade the environmental pollutants, relying on observation and trial and error for effective environmental cleanup. Now, these old traditional methods, which in many ways are rudimentary, actually provided a lot of foundational insights into the principles of microbial processes and paved the way for development of cutting edge technologies, innovations and applications. So, due to these developments in these different aspects, we have today very advanced techniques for different applications. So, some of the advancements are the genetic engineering, synthetic biology and modern applications of gene editing technologies like CRISPR-Cas9 and also the possibilities of making synthetic genomes. Let us now discuss about some of the traditional microbial biotechnology methods and the various products which are produced by these processes. So, for example, in the case of traditional fermentation, it is one of the known earliest applications of biotechnology, particularly microbial biotechnology, and its origin traced back to thousands of years. These traditional fermentation processes utilizes the natural metabolic activities of microorganisms such as bacteria and yeast to transform raw materials into valuable products. So, some of the simple products, for example, this is a traditional Oriya dish called pakhala. This is prepared by soaking leftover rice in water for mild fermentation overnight. So, this is also a way of utilizing leftover food so that there is less food wastage. And these similar kind of fermented food rice is also found in West Bengal, where it is called as panta bhat, and also in Assam called poita bhat, and other states like Bihar and Jharkhand, where it is called basia bhat, and also in South India, Tamil Nadu, where it is known as kanji. Now, modern research is trying to find out many of the beneficial effects of these kind of fermented rice, and it has been reported to help in maintaining the gut health to a large extent. Now, there are other products of traditional fermentation like beverages, beer. So, here the cereals are fermented by yeast to produce these alcoholic drinks, and this is one of the oldest known biotechnological practices, and many ancient civilizations including the Sumerians and Egyptians mastered the art of brewing. Yeast ferments sugar into ethanol and carbon dioxide, resulting in beer with various flavors and alcoholic content. So, another very old technology, but which is still used in mass scale, is the making of bread, where yeast fermentation is used in bread making. This is also another ancient process. Here, yeast causes the dough to rise, creating a light and airy texture. Traditional methods involve the natural fermentation processes that were optimized over centuries for better bread quality. So, here in this picture you can see these very large vessels used for fermenting and storing beer in a brewery in Jajjak. Now, there are other kind of fermented food which are produced by these traditional methods of microbial biotechnology. For example, the production of yogurt and cheese, the fermentation of milk to produce yogurt and cheese is a significant development in food biotechnology, whereby the shelf life of milk can be increased and the food products can be stored for long time. In such case, bacteria like lactobacillus and streptococcus are used to ferment lactose in milk, producing yogurt, while different bacterial and fungal cultures contribute to the flavor and texture of cheese. These processes were developed through empirical knowledge and have been refined over generations. There are also other fermented foods across the world known to various cultures and civilizations. For example, you have fermented vegetables like sauerkraut and kimchi, and these have been the staple food in countries like Korea, Germany. Here also lactic acid do fermentation of the sugars. For example, in the case of sauerkraut, the sugar is fermented from cabbage, and this helps in the preserving and enhancing the nutritional value of these vegetables. So, these traditional fermentation techniques contributed to the development of probiotic foods, promoting the health and nutrition of the consumers. Another area which we have mentioned is early bioremediation efforts or technologies. So, here the microorganisms were used for environmental cleanup, and particularly this took shape in the latter half of the 20th century. So, here you can see that petroleum is getting spilled into the sea, and these petroleum can be degraded by microbes like Alkanivorax. So, they particularly metabolize the hydrocarbons and convert them into carbon dioxide and water, which are harmless. So, this finally helps these microbes to thrive in these oil spill areas, and it plays a crucial role in reducing the environmental pollutants. So, in the 1980s, these became very important areas of research, particularly for the technology development of oil spill cleanup. So, this happened because of the environmental challenges that happen due to oil spills. So, you can see here in these pictures there are certain wells. For example, there is an injection well, there is a monitoring well, and there is an extraction well. So, monitoring well helps us into looking into inside of the wells of the various happenings. So, you can see here again the oil is getting spilled and then contaminating the groundwater. Now, these groundwater is not suitable for human consumption. So, this has to be made free from these oil spills. So, in this case naturally occurring microorganisms along with nutrients and oxygen are injected into these wells and they land up in the oil spill, where these microorganisms carry out the degradation of these pollutants. And then this extraction well helps us in pumping out the byproducts of these reactions, and they are again pumped back into the oil well, so that any residual pollutants can be further degraded by these microorganisms. So, in brief, these kind of technique is known as in situ bioremediation, which means we are doing the bioremediation on the site of the oil spill, and this involves injecting gases, nutrients or microbes into the soil to remove pollutants, which basically takes care oil spills from leaks or buried waste that contaminate the groundwater. And then the injected oxygen helps in detoxifying the pollutants, converting them into carbon dioxide and water with the help of the microorganisms. There are certain challenges and limitations, of course, in this entire process, which occur due to our limited knowledge of the microbial capabilities on the one hand, and the environmental variability on the other hand. But now with research, we have been able to make these processes much more efficient. For example, they are now much more efficient strains, and there are efficient processes due to which the biodegradation utilizing microbes has become cost effective and beneficial. Now, with regards to oil spill pollutant degradation, we have to discuss the initiatives and pioneering work by Professor Anand Mohan Chakraborty, who is often considered as the superbug superhero. So, in 1971, Professor Chakraborty developed the first genetically engineered oil eating bacterium, which we call as the superbug, particularly from the species Pseudomonas putida. So, these particular genetically engineered Pseudomonas was capable of degrading 2/3 of hydrocarbons found in typical oil spills more efficiently, paving the way for modern bioremediation technologies. So, for his pioneering work, Professor Chakraborty is considered as one of the most brilliant microbiologists, and he was the first to secure the US patent on a living organism back in 1981. His groundbreaking achievement paved the way for the legal ownership of genetically engineered organisms, sparking a wave of biotech innovation and commercialization. We will speaking about the IPI issues in one lecture in probably module number 12, where we will be discussing about Professor Chakraborty's work in more detail. Another advancements in the area of microbial biotechnology is that of antibiotics. In fact, the modern era of microbial biotechnology is said to have begun with the discovery of antibiotics by Sir Alexander Fleming. So, in 1928, Alexander Fleming discovered penicillin. This was the first antibiotic produced by any mold, particularly in this case Penicillium notatum, and these breakthrough revolutionized medicine by providing an effective treatment for bacterial infections. Now, let us look into some other important discoveries, inventions which played a role in shaping modern microbial biotechnology. So, as mentioned, the work by Alexander Fleming in 1928 is considered a pioneering event in the history of microbial biotechnology, followed by another similar discovery of streptomycin by Waxman. Then we already discussed about the superbug developed by Professor Chakraborty in the 1970s. Around the same time, the discovery of restriction enzymes happened, and which helped us in cutting out desirable genes from organisms. And then other developments like the vectors and ligases helped us in doing recombinant DNA work. So, by around 1972, the development of recombinant DNA technology by Pohlberg and colleague is another important milestone in this historical journey. And the creation of genetically modified bacteria by Herbert Boyer and Stanley Cohen in 1973 is no less than a science fiction and an important event in this journey. Then we have other important developments like the development of techniques for genetically modified plants, although these are not directly related to microbial biotechnology, but then it is a microbe, Agrobacterium tumefaciens, on which these technologies are based. So, these are one of the most important progress in this field. Then one of the most cutting edge technology which has so many different kind of applications is that of polymerase chain reaction developed by Kary Mullis in 1983, and then this has now today evolved to many different forms including RT-PCR, which is used extensively in the detection of infectious diseases like COVID. And then we have the advancements in the use of beneficial microbes for probiotics and fermentation, along with the advent of metagenomic analysis in the 1990s, and the advances in the high throughput sequencing technologies and development of microbial fuel cells for sustainable energy production happened something around 2000. And then the creation of synthetic bacterial genomes by J. Krieg Venter Institute in 2010, almost it was something like playing God, trying to create life forms, of course not totally from scratch. In this case, genes and constructs were stitched together from different sources, but anyway, this was one of the most remarkable achievement of this century. And of late, the genome editing technologies by Jennifer Doudna and Emmanuel Charpentier is revolutionizing the field of microbial biotechnology. So now with these introduction and background, let us try to understand the various principles involved in the field of microbial biotechnology. Mostly we will be discussing about the microbial diversity, the genetic manipulation, the fermentation technologies, bioprocess engineering, metabolic engineering, biotransformation, environmental applications, microbial production systems, quality control and safety, and finally the sustainability. So we have already mentioned that microbial biotechnology leverages the microorganisms to develop products, number one, processes number two, and offer solutions across various industries like agriculture, medicine, and environmental management. And in these, several principles guide the application of microorganisms to solve complex problems and create innovative solutions across various fields. So let us study about the microbial diversity and how it is important in microbial biotechnology. To exploit the vast diversity of microorganisms ranging from bacteria, fungi, virus, and also archaea, to discover and utilize unique metabolic pathways and enzymes, it is very important to have idea on the diversity of these microorganisms. The second most important aspect is of genetic manipulation, where we have two important technologies. One is that of gene cloning, where we insert specific genes into microorganisms to produce desired products. For example, the production of insulin and several other vaccines are saving millions of lives at today's date. Then genome editing technologies, as one of the technologies CRISPR-Cas9, then there are other two technologies, ZFN and TALEN. So these technology enables us to make precise changes in the microbial genomes for enhanced traits or functionalities. The next is the fermentation technology, where we use microbial fermentation processes to produce chemicals, pharmaceuticals, biofuels, and food products. And by optimizing the conditions, we can maximize the yield and efficiency. So we can see that microbial biotechnology offers a huge plethora of solutions as regards to industrial production. So in bioprocess engineering, we go for the designing and operating reactors for scaling up. For example, for producing insulin for a entire world, we need the help of bioprocess engineering, where we can use microorganisms to be cultivated in huge fermenters to grow and produce targeted products, like say for example insulin or certain vaccines. So this requires scaling up from lab to industrial production, and is one of the most important aspects in this particular technological process. Another method is principle is that of metabolic engineering, which we use for modifying microbial metabolic pathways to enhance production of target compounds or degrade pollutants. So today, with the power of genome editing, we are able to modify these metabolic pathways in much more efficient way, and compared to what we could achieve with simple gene cloning technologies some decades back. So in these kind of approaches, we try to balance the metabolic fluxes to optimize product yield and reduce the byproducts. Biotransformation is another important principle. So here we use microbes to catalyze chemical reactions, converting substrates into valuable products like drug synthesis, environmental detoxification, etc. And now the principles of environmental applications are also very important, and we have already spoken about traditional bioremediation process, where microbes are used to clean up the environment from contaminants like oil spills, and it is also possible to clean up the environment pollution which have heavy metals. Then other important pollutants today are the biopesticides, which are used for raising crops. So after the crop is harvested, these residues remain in the soil or it goes into the water bodies. So we can use microorganisms or the products as natural pest control agents in agriculture, which we know as the biopesticides. Another important principle is that of microbial production systems. Particularly, we are interested about cell-free systems, where we use extracts or purified enzymes from microbes rather than the whole microbial cell for industrial processes, avoiding the need to cultivate the live organisms. And then also the whole cell systems, where we employ the live microbes for continuous production or degradation processes. Then comes the quality control and safety principles. To ensure the safety, consistency and efficacy of microbial products through rigorous testing and compliance with regulatory standards, we need to address potential risk associated with genetically modified organisms and as well as the microbial pathogens. Finally, the principle of sustainability is supreme to all of these, because whatever processes we develop to minimize waste and energy consumption, we should always try to maximize the efficiency and the economic viability of these applications, and therefore we need to promote the use of renewable resources and eco-friendly practices. Let us now discuss about the contributions of microbial biotechnology, particularly the advancements of biotechnology and impact on related disciplines, and its use in human and animal healthcare, and finally the integration of the various disciplines. First, the contributions of microbial biotechnology. So, we can see that the advancements of biotechnology has helped us in the industrial applications. Particularly, it has revolutionized the production process through engineered microbes, such as those that produce enzymes, then biofuels, and also today bioplastic production is becoming a very important area. Then next is the pharmaceuticals. Here the production of antibiotics, vaccines, therapeutic proteins and is facilitated, along with the improvement of drug efficacy and finally the affordability by cutting down the costs. Then other things are like the biotechnology innovations, particularly the advancements in gene editing, then synthetic biology and several other biotechnological fields are driving the advancements in the microbial biotechnology. So, the impact of microbial biotechnology spans across industrial processes, environmental sustainability and healthcare, offering innovative solutions to global challenges and improving the quality of life for human and animal alike. In the case of environmental science, we have enhanced environmental management through the bioremediation and waste treatment. In agriculture, it supports sustainable practices like providing inputs such as biofertilizers, biopesticides and microbial inoculants. In the case of food industry, it contributes by improving the food quality and food safety through fermentation and preservation technologies, and its impact on human health include the advances in microbial biotechnology contribute to the development of novel vaccines, probiotics and therapeutic agents. For example, the recombinant DNA technology enables the production of human insulin and growth hormones significantly, impacting treatment for diabetes and growth disorders. So, if we consider the old method of insulin production, basically it was prepared from the calf thymus, for which the young calves have to be sacrificed. So, for producing a few grams of insulin, thousands of calf has to be sacrificed. Now, along with it, if we calculate the cost of land required to raise those calves, their parents, so the cost is high and the viability actually reduces. So, thanks to advancements in recombinant DNA technology, which now enables us producing human insulin in large amounts in a small factory. Now, when it comes to animal health, the impact is somewhat similar. The animals also benefited by the development of vaccines, probiotics and antibiotics, which improve their health as well as their productivity. And the microbial technology helps in creating vaccines that protect livestock from diseases and enhances the nutritional quality of the animal feed. Now, let us discuss about the integration of disciplines vis-a-vis microbial biotechnology. So, we can see that microbial biotechnology is impacted by fundamental research, and it is always dictated by practical applications. So, the interaction of these two through the integration of various disciplines finally leads to the advancements in biotechnology and other related fields. So, if you take a few examples like that of genetics and molecular biology, so here you utilize the gene editing and recombinant DNA technologies to modify organisms for various applications, some of which we have already discussed, including designing metabolic pathways. And then if we look into other disciplines like biochemistry and chemical engineering, which involves the biochemical manipulation of microbes and optimization of the production processes. So, these helps us in cutting down the cost of products and also make the production in large volumes, which is scaling up of the products, along with ensuring that the products are safe and they comply to the regulatory guidelines of various authorities. So, in brief, microbial biotechnology bridges fundamental research with practical applications and contribute to advancements in biotechnology and the related field. Now, with this background, we will be discussing some of the emerging hot topics, and some of these are already making impact in the field of microbial biotechnology. So, we have discussed a lot about gene editing, which is revolutionizing biotechnology. We will also in future artificial intelligence is going to play a big role, similarly synthetic biology, and one of the most important problems of our time is that of climate change. So, microbial biotechnology is also going to help us mitigate some of the climate change challenges. So, if we look into the various research that is happening, so we can see that people are now interested in accurate microbial genome editing technologies on the one hand, and then also they are interested about using artificial intelligence for microbial biotechnology and use synthetic biology for microbial community design. So, these are some of the important developments which is setting the agenda for the future. So, we can see that microbial biotechnology is advancing rapidly, which is driven by the innovations in genome editing, artificial intelligence and synthetic biology, and climate change is demanding eco-friendly solutions to emerging challenges. In this context, these tools are unlocking new possibilities in medicine, agriculture, industry and environmental sustainability. So, we will discuss briefly how these innovations and climate considerations are shaping the future of the discipline. For example, in the case of genome editing, which we have already discussed to some extent in the earlier slides, one of the advantages of these genome editing technologies, the CRISPR-Cas9 system, is the precision and customization. So, these tools allow precise modifications to microbial genomes, unlike the old technologies of gene cloning. So, these has enhanced the ability to address climate change related issues. For instance, we can use the engineered microbes to design carbon capture or degrade the pollutants in the environment much more efficiently. Another issue is that of biocontainment and biosafety. So, today we are interested in developing microbes which are known as the climate resilient strains. Even if there is some change in the climate, the temperature goes high or low, these microbial strains will still be equally efficient in producing the products that we are interested in. So, this can be created with gene editing, with built-in safety mechanisms, ensuring that genetically modified organisms will not adversely affect ecosystems as they are deployed to tackle climate related challenges in the field. The third most important aspect is that of functional genomics, where a lot of focus is there on the climate adaptation, where gene editing can reveal and modify microbial traits that enhance resilience to climate induced stresses such as temperature fluctuations, drought and salinity, contributing to more robust microbial applications in agriculture and environmental management. The another important discipline which is bringing in a lot of promise to microbial biotechnology is that of artificial intelligence, particularly in the area of data analysis and prediction. When it comes to climate modeling, it is a very complex subject, and artificial intelligence can analyze vast data sets from climate studies and the microbial interactions to predict how microbes will respond to climate change, enabling the development of more effective strategies for using microbes in climate mitigation and adaptation. In this regard, one important aspect is that of the melting permafrost, where due to climate change, where it is thought that as these permafrost melts, a lot of microbes, unknown microbes which was trapped in the past will actually get released into the environment. So, from that aspect, artificial intelligence will help us in dealing with these unknown microbes, some of which may be actually harmful and some of them may be beneficial. So, another thing is issue is the optimized environmental solutions, where artificial intelligence will be used for optimization of microbial bioremediation processes, to improve the efficiency of microbes in degrading pollutants, for addressing the environmental impacts of climate change, such as increased frequency of oil spills and chemical pollution. Over and above these, one interesting aspect is that of personalized solutions. So, for example, the case of climate resilient probiotics. Now, there is a lot of hype in this field, and there are discussions whether artificial intelligent tailored probiotic strains to individual needs is a possibility, and then whether it can enhance agricultural productivity and soil health in the face of the changing climatic conditions. Another area that is contributing to microbial biotechnology is the synthetic biology, particularly in the design and construction of novel biological systems, and with respect to climate adapted microbes. Synthetic biology enables the creation of microbes with engineered traits for climatic adaptation, such as enhanced drought resistance or increased efficiency in carbon capture and storage. Then comes the biological devices and systems, where climate monitoring tools are very important, and here synthetic biology can produce novel biosensors and biological devices that monitor climate related parameters such as atmospheric carbon dioxide levels or soil moisture, providing real time data for climate adaptation strategies. And finally, the bioengineering and systems biology approaches are also going to be very important, where we are looking for integrated solutions through synthetic biology combined with systems biology for designing microorganisms that perform complex functions like carbon sequestration or degradation of greenhouse gases, contributing to climate change and mitigation efforts. Now, if you look into various climate considerations and environmental management overall, microbial biotechnology plays a very central role here. For example, in mitigating greenhouse gases, in maintaining healthy soil and water, and there are also concepts of circular bioeconomy, where the waste of one enterprise become the input for another enterprise, thereby closing the loop, and there will be no any waste which will be dumped, and there is a full utility of resources. And this kind of approach is known as the circular bioeconomy, and this would help creating new enterprises and thereby create new products and new for new markets, as well as it also addresses the problem of social problems like unemployment. So, in this entire scenario, microbial biotechnology is going to play a crucial role, because using waste from one enterprise as inputs for another enterprise, we need to modify microbes utilizing various approaches like synthetic biology or gene editing, and artificial intelligence is going to help us in this entire process. Now, whatever microbial processes, the natural ones or engineered ones, we use in this entire process of adaptation to climate change or mitigating climate change, we have to ensure sustainable practices all throughout the process. So, in the entire scenario, microbial biotechnology is going to play a crucial role. If we once again focus on pollutant degradation, which we have already discussed at length, we can see that microbial biotechnology provides solutions for mitigating climate change by developing microbes which are capable of degrading pollutants and toxins from industrial activities and climate related disasters. Carbon sequestration with engineered microbes is a possibility which will reduce atmospheric carbon dioxide, one of the key drivers of climate change. Besides these, other greenhouse gases are also challenges, and microbial biotechnology can help us in mitigating many of such challenges. And one important aspect is that of making agriculture sustainable. With our increasing global population, the demand for food is increasing, while the number of people who goes hungry to bed every night is also similarly increasing. So, there is a need for enhanced food production, but for this we cannot go and claim more land by converting the forest land into agricultural land, because that will make the climate change challenge much more aggravated. So, for this, microbial biotechnology is going to play a key role by enhancing the soil health and fertility, promoting climate resilient crops, and reducing the resilience on reliance on chemical fertilizers, and contribute to greenhouse gas emission mitigation. So, with this we come to the end of this first lecture. Thank you for your patient hearing.